Good afternoon, everyone. We are Biomedical Science 1 from grade 12, room 7 p.m., and today we will be tackling the possibilities of gene editing. Before we formally start with the presentation, we first have to tackle or know what gene editing really is. So, what is gene editing? Genome editing, also known as CRISPR's gene editing system, is a very powerful tool for genomic research that targets specific locations in the DNA with its given components DNA and RNA. Furthermore, it is a type of genetic engineering in which DNA is inserted, deleted, modified, or replaced in the genome of a living organism. CRISPR gene editing creates the possibility of correcting errors in the DNA, and one of the key significance of this method is that it allows the researchers to easily alter DNA sequences and modify gene function. For our claim, CRISPR gene editing is susceptible to various side effects. Based on the article by Cyranowski entitled "The CRISPR Baby Scandal: What's Next for Human Gene Editing?" It is stated there that some are concerned about how the public scrutiny will affect the future of the field, specifically the medical field, whether or not researchers aim to alter the germline. Furthermore, the negative focus is, of course, not good. Says Frederick Lanner, a stem cell scientist at the Karolinska University Hospital in Stockholm, who has been editing genes in human embryos to study how cells regulate themselves. Furthermore, since the CRISPR gene editing has been proven that the amount of scientists who have tried this method is not yet sufficient, it is really prone to numerous dangerous side effects both on the child and the mother as well.
The group believes that the most applicable paradigm would be the post-positivist, since the chosen facet requires a deep knowledge regarding the possible side effects of the gene editing. According to the University of Chicago Press journals, a post-positivist research approach advocates methodological pluralism and is based on the assumption that the method to be applied in a particular study should be selected based on the research question being addressed, which allows the researchers to be able to record according to the probability and objectives.
The group recommends the use of a qualitative approach for this research. Some research methods that the researchers can use are interview, focus group discussion, and observation, with the researchers' intent to understand the possible side effects of CRISPR when used in gene editing. The use of a qualitative approach, which comprises of diverse perspectives and practices, would be the best approach to use. And so, in order for the researchers to have these different perspectives and practices, they will need to interview, observe, and have a focus group discussion with different professionals that are well informed with CRISPR and are continuously studying it today. Now that we know that CRISPR gene editing is susceptible to various side effects, let us always keep in mind to always think if something is beneficial before actually doing it. That is it for now. Thank you all so much for listening to our presentation and have a great day ahead.